Fluorescent proteins were first discovered in the 1960s by a group of scientists that were just interested in animals that glow, animals that produce their own light. So bioluminescence is really a compound of fuel. There's a luciferin and a luciferase. When you add them together, you expend this energy and you produce light. And they were going around trying to find different luciferin, luciferases and study just for pure scientific knowledge. And in doing so, they noticed that one of them, when they were able to purify it, it was coming up with, it was in the organism itself, in the jellyfish, it was green. But when they purified the components, it was blue. So they realized there was something there that was changing the color from blue to green. And that was this other contaminant, this fluorescent protein. Recently, we decoded the human genome, and that shows you that we have about 25,000 different proteins genes that we're able to make. But how they're that gives you more of like a phone book. So if you're trying to study a city, and somebody hands you a phone book, and they say, tell me how the city is interacting. How are the people interacting in the city? And you're left with a big phone book directory. And what this protein allows you to do is allows you to see all the cells moving around in a living organism. You could put this reflector in front of any protein and watch it interact, who it's talking to, what happens in diseases, for instance, when things start going wrong. You could also put little selective tags in to assassinate proteins whenever you like. Therefore, you could blow up a protein and see what happens when you take a member of the society away. So in just a short time, this was in the 1990s, that a professor put this into a transparent worm. And since that time, the research has really burgeoned. There's been tens of thousands of papers using this small jellyfish protein. I've assembled a team of medical scientists and physics and marine biologists, and we have embarked on several worldwide expeditions to coral reefs. During the day, we scuba dive on the reef, looking for unique corals that we've yet to examine. And once we've identified them, we'll pop up one of these glowing light sticks. Looking at the fluorescence could only be done at night. So we first follow the light sticks back to the portion of the reef that we'd placed these earlier, and then we drop down now. And we kind of find our way along the reef until we see the area that we were working in. And at that point, we'll turn off our lights, and you'll see that we're just shining one specific beam of light here. And in this instance, we're shining a blue beam of light or a violet beam of light. We're looking at it with a camera that has a green filter on it. So we could see through the green filter these are living coral polyps that are actually glowing. Here are some examples of patterns that show the different fluorescent proteins in corals. You could see that in some cases it's clearly targeted to specific anatomical regions. At Baruch College, we're actually funded by the National Science Foundation, and this summer I'll be taking students both to the Little Cayman Island and to the Red Sea and to the Mediterranean, which we'll be examining on expeditions this summer, as well as a new field course, Tropical Reef Ecology, that we'll be running for the first time in January 2011. So we'll be bringing 15 Baruch students to Little Cayman Island, which is one of the smallest of the Cayman Islands. It's only about 100 people live there, and I'm affiliated with a marine station on the island, the Central Caribbean Marine Institute, which has access to one of the most spectacular coral walls in the Caribbean, the Bloody Bay Wall.